Morning, everyone. Thank you all for being here, and thank you to the Chamber of Commerce for affording Aldevron the opportunity to speak a little bit about what we do. Aldevron is there we go. Aldevron is what's known as a contract development and manufacturing organisation. In the biopharmaceutical world, we fill the role that is often an element that the smaller biopharmas and even some of the larger caps now don't want to do, which is the development and the manufacturing. They prefer to put their efforts into their clinical work and then onto their R&D side. So we're sort of an extension into their world. We have a portfolio with several things in it. The primary ones are showing up there. We have a monoclonal discovery group. We have protein and enzyme manufacturing, including things like gene editing enzymes. We have plasmid DNA, which is far and away still the largest component of what we do, and some subclasses of plasmid DNA. And then we have RNA as well, a couple of different types of RNA that we manufacture. Plasmids are sort of the unifying string of all the biopharmaceutical and biotechnology industries. At some point along the way, in some product, you will be manipulating genes, you will be transferring those things that the genes encode, and so they are a broadly utilized tool in the industry. And what they are is they're little loops of DNA that replicate inside bacteria, and some other microorganisms, but primarily bacteria and primarily a few specific types of bacteria. And so essentially when you grow up a lot of bacteria, you grow up a lot of plasmid DNA, and you need to be able to purify that out. It makes up about 1/500th of the mass, and so that's where some of the art lies in this. We didn't actually start out as a CDMO. We started out as two guys straight out of college. You see, Mike still looks the same, and I still have the same shirt that I've got in the bottom there. But the two guys out of college, and we had this high-minded idea of utilising plasmids as a way of helping to counteract or create a countermeasure for antibiotic resistance, particularly in Staph aureus. And it's still a damn good idea, and it's still something no one's been able to do. But when you're wandering around trying to convince people to give you a bunch of money to go and maybe do something that could possibly work, it's a bit of a hard sell. And so we came to the realization that, ramen noodles are fine, but they're not that good, and we needed to get off our ass and actually do something. And one of the things that we'd been working with, obviously, were plasmids, as they were going to be the crux of the base of this technology. And we understood that there are a lot of people using them, and the ability to make them in relatively small quantities had been commoditized, and you could do it in your own lab. But it was a lot of work, and it required a lot of dedicated equipment, and it was something that people did not want to focus on. So there's Victoria. Is that Victoria coming in? I thought it was. So we decided that we would create a business plan. It was more as a rite of passage than as a funding vehicle, as it turned out. But Mike still has a copy of that, and it is one of the most eerily clairvoyant things you will ever read. Everything we said that we were going to do happened, and we actually did it. It's sort of an ancient tome now that we found in an old filing cabinet, but it goes to show that at the very least we kind of had an idea of what we were doing. We weren't able to convince many other people that we did, so we sort of had to start off a pretty hard scrabble, as I know a lot of businesses start. It was no different for us. Actually, I'll just go back one there. So one of the things that isn't perhaps reflected on this so much are some little sorts of inflection points that happened along the way. But two that are important to note are the acquisition of the monoclonal asset in in Germany, in Freiburg, and then the protein group in Madison, which was an ex-Novogen operation. And the reason that's important is because both of those things overlap with the scope of DNA. When I was talking before about it being a unifying string, that's an important thing to note, that all the things that we work with are related. And so they have a broad footprint across the industry that gives us a lot of touch points. You know, the progress along the number of employees, I'm not sure how accurate that is at the far end now. It's a few more than that now. But, you know, along the way, among the milestones that happened for us, or events that sort of happened for us, in 1999, not long after we'd started, a young man named Jesse Gelsinger was the first person to die as a result directly of a gene therapy being administered. He had a somewhat mild form of a rare disorder, and he died about 4 days after being dosed. Now, that cast a real pall over the industry and, you know, gene therapy in general. And some of the other things that were going on was the sort of failure of plasmid DNA to really manifest itself at that point as a frontline tool for a vaccine or a gene therapy. A lot of the stuff that we'd shown in mice didn't work when you went higher and higher up the food chain, as it were. And so a lot of our competitors, although they would never have considered us competitors at the time, they wouldn't have found Fargo on a map, frankly, but they had gone ahead, they'd rushed ahead, and they'd built the shiny palace with the $10 million ferment in it and all the rest of it, waiting for the clinical manufacturing to come to them, and it never came. We, on the other hand, just stayed and did what we did. We worked with researchers at universities, got to know what they were doing, we got to know the programs, etc. So we weren't exposed to that massive boom or bust cycle because there was always an underlying element for use of the DNA. We started off in Van Ayers Hall, and we sort of matriculated to Holtz Hall, and that was interesting, having Madagascan hissing cockroaches as neighbours to a manufacturing lab. Thankfully, the FDA never came by. And then in a major inflection point, we moved down to a building that we still use. We call it Main. It's behind Hornbachers on 32nd Avenue. And that was an incredible break for us that Tom Lavelle and Grant Johnson, our landlords, did a fantastic thing for us to allow us into that space at the rate that we were paying at that point in time. That was of major importance to us, and I know neither of them are here today, but those are the sorts of breaks and things that happen along the way that really allow a young nascent company to take off. So if you look at the by time a number of clients, you know, it's a bit misleading because each of those clients could be doing 20, 30 jobs with us in a given year, and some of them may be producing grams and grams of DNA, and some of them only a small amount. But as you can see, in about the mid-2000s, things really sort of took off. The number of clients, our scope of capabilities, our professionalization of marketing and accounting. We had no idea what leverage debt was at that point in time. We know what debt was. We didn't know what leverage debt was. We didn't know what EBITDA was. We didn't know any of this cool stuff. Well, some people think it's cool, but you know, in one of the points that really you know, people say, well, you know, you guys were at the right place at the right time when the industry took off, and yeah, that's true, but not a lot of the people appreciate how much time it took you to be at that place, right? How much work, how much effort, getting knocked on your ass, getting back up again, et cetera, that you have to do to survive in the game. And you take your lumps, you move on, you get smarter, but you always, always, always understand what the client is working on and what's important to them. To be as collaborational as possible in our game has always been one of our strengths, and it was formed not just around our character of the individuals in the company, but by necessity, needing to innovate. And it's in that innovation really that at the end of about 2007 to 2008, the virus that had killed Jesse Gelsinger was no longer being used, or was very rarely being used as a vehicle or a vector of gene therapy. Researchers had been working with much milder and safer forms of viruses, one of them a derivative of HIV, and the other one called AAV. And at that point in time, the only way, and these were still sort of drug non grata, as it were, the only way that you could work with these viruses was to make them with plasmids, right? You needed to take all the different parts of the virus, put them into a cell, and make the virus that would then either go into a person's cell to do its thing, or it would go directly into a person. And the only way to make that DNA was under what is called GMPs. The FDA, EMA over in Europe, have guidances which are very strict as it relates to how you can make drugs. And there's a very good reason for that. But biological drugs are incredibly costly to develop, and funding in this area was very, very low at that point in time. The whole industry was hanging on by a thread. In order to matriculate these things through to the coalface where they were needed, it needed a more cost-effective alternative to full GMP manufacturing. We'd never touched clinical at that point in time because we were scared of what we'd seen in the past, and we thought we knew our space very well, and we were keen just to stay in there. One of the sort of foremost viral shop guys around came to us and said, look, this industry is going to die unless we can find a way to make this stuff cheaper and faster to get these clinical trials rolling. What can you guys do? And so we sort of threaded the line between the nimbleness and science of what we knew we had to do and the sort of regulatory shroud that you wrap around a manufacturing process, and came up with a new quality level that we call GMP source. GMP source being, it's the source for making the virus, it's not the stuff that goes directly into the person. And the FDA bought off on it. And it's impossible now to think about things that are on the market that would never have come to be if it hadn't been for that meeting with that particular guy that made us go down that path. And it totally changed the trajectory of the company and actually of drug development in that space. You know, our mission is pretty clear. When we succeed, our clients succeed, vice versa. We get jobs through, we get more jobs in. You keep feeding the machine, you get more cures out there. The image on the right is of Carrie, Emily, and Tom Whitehead. I forget the dog's name. I did know it at one point. A few years back, Emily was diagnosed with acute lymphoblastic leukemia, which is an absolute death sentence. Her parents had been told to say goodbye, and she was put into more or less terminal care. And then a trial that had been opened, that was running parallel with this, she was entered into. And she became the first child, and I think it was the third or fourth person to receive what we now call CAR T therapy, and what is now the drug Kymriah, Novartis drug. And it saved her life. She's no longer in remission. She is cured, technically, which is a term that oncologists hate people to say, but it's becoming more and more part of the vernacular. If you ever get a chance, look for Emily Whitehead or Tom Whitehead online. You might find Tom's a pretty tough guy, and it's a pretty tough talk that he gives to listen to, but at least you know there's a happy ending. It's well worth looking at. And Mike knows the family very well. He was at an event with them a couple of years back, and a clinician came up to him and said, you know, the work that you guys are doing has saved a kindergarten's worth of kids so far. And it's probably more like a junior high's worth now. But again, that's an incredibly, things like that, success stories like Luxturna for blindness, are the things that drive us on. And wow. Okay, I guess I'm, all right, well, we'll hurry through the next bit. It was flashing, it looked like about to blow up or something. Okay. So our clients are breaking new ground all the time. They're at the highest risk edge of the game. We always say they're at the bleeding edge, the leading edge. Many of them are in a space where the risk-reward ratio is so in favor of reward because there are no alternatives that we have the ability to influence how these things are shaped and how they enter the market. As I say, we innovate, we advance. We've had to innovate. That was part of our DNA, as it were, by necessity. We collaborate. The instance with the researcher in the virology space is one. There are many, many other instances. And we make lives better and we serve our clientele. I think that's a generic statement, but it's very factual in our instance. Well, we've got some nice pictures of places where if you ever get down to 40th Avenue, you can see that one. If you ever get to Madison, you can see that one. It's not that exciting. If you ever get to Freiburg in Germany and you're driving around lost, and you see those two lions, it's possible that it's our place, but they've got quite a few lions over in front. Obviously, we have a lot of spaces around town. As we grow, our growth has sort of mimicked the area in a way. A lot's changed in the last 21 years since we started. The flow of information and ideas has minimized the tyranny of distance to a great extent. There is a critical mass now in this area, a gravity that we have. We used to orbit Minneapolis. Now we have towns orbiting us in this space, which is great for our industry. The more tech industries in the area, the better. So that's 40th Avenue, I think, and that's 41st Street. I'm sorry about the speed I'm going there. And then we're going to have an admin building, and as I saw when I was looking through these slides, we're only going to use silhouette people from now on to lower our carbon footprint going forward. Yeah, there's a couple more of them. And so our campus build-out is in part, people might not understand. The folks at Hector, I should get free flights because there are about 100 or more groups a year come in here from all over the world to audit, to meet, et cetera, et cetera. And some of them send what's called a PIP, a person and plant, to watch a manufacturing process. So we need to adjust our facilities, not just to have more and more space to grow things and purify things, but to support our R&D efforts, our training, and our client experience. So I think, there might be a couple more silhouette people. No, done. Okay, yeah, I better stop there. And so, Tiffany, you got to speed up by 5 minutes to get us back on time. Thanks.